Topic is navigating drug development to target endoplasmic reticulum in neurodegenerative diseases. So, I am Dr. Tanzeer, I am coordinator of this Gyan course. So, now what is the journey of a drug development? How does a drug development starts? So, there are 5 stages for drug development. The first is that first of all we should know about the disease. We should know its molecular mechanism, and from that molecular mechanism we find out the targets. So, the first step of any drug development is finding a target, selecting a target. So, after we select a target, and it is not easy, as Alan has shown you so many molecular mechanisms of diseases. Now, you know that it is so complex, many diseases are so complex, we do not have a single target in one particular disease. That is why we do not have cures for neurological disorders, we do not have cures for cancer, because the mechanism is complex. But as we will do this basic research, as we delve deeper in the molecular mechanism of these diseases, we may come up with some targets and then design drugs against them. So, the first step is selecting a target for which we need to develop a drug. Then second step is lead discovery. Now, you know a target, your target can be an enzyme, it can be a receptor, it can be a protein, it can be a DNA. On the basis of, it can be a carbohydrate, it can be anything. On the basis of the type of molecule that target is, you select a molecule, a lead molecule which could bind it, either enhance its property or decrease its property. So, the second step is lead identification or lead discovery, finding a molecule which can modify the target and thus impact the disease progress. Then comes the third step in drug designing, that is medicinal chemistry. Now, we can find any lead molecule which could modify your drug target. Just a minute, एक मिनट आप इधर से आना पीछे से. So, we have found a lead molecule which could modify that particular target, but then we see that how much effective is that lead molecule, and then we work on improving its efficacy, improving its properties. It should be good. I mean, if we give this particular molecule to a human being, it should have a very good absorbance. It should actually reach the site of target, and it should have a very good efficacy at very less dosage. So, then comes this third step, that is medicinal chemistry, where we enhance the activity of your lead molecule. After we have developed a molecule which is very good, which is having all the properties to become a drug and is acting on your target, then we take this lead molecule, this modified molecule, and test its activity in vitro and in vivo. So, till these 5 stages is the preclinical stage. So, after preclinical studies, then we take the molecule, the final molecule which has passed all these levels, to the clinical trials. And believe me, after doing so much of research, after studying number of molecules, when we take them to the clinical trial, most of the drugs actually fail in clinical trial. So, this drug development is a very cumbersome field, and sometimes 1 in millions of molecule could actually reach the market and could actually reach the patients. So, now we will talk about target selection in neurodegenerative diseases. Okay. So far you have seen so many different mechanisms. Alan is mostly talking about mitochondria, phagy. So if we see, there are many, because we don't know the main reason how it initiates. So we have to pick something in between, we have to pick anything in between which we think could be a step which is at the very beginning of the disease progression as our target. So when we see all these diseases, let us see Alzheimer's, Parkinson's, ALS, Huntington disease, every neurodegenerative disease have their own hallmarker protein. Like in Parkinson disease, it is alpha synuclein, which is a hallmark protein, and it aggregates, form Lewy bodies and all that. In Alzheimer's disease, it is amyloid beta, tau protein also. They are the reason, they are the hallmark proteins, they are the reason for the progress of these neurodegenerative disorder. ALS is superoxide dismutase 1, this is a copper zinc SOD, and then TDP43. Then in Huntington disease, there is a protein Huntington. So all these neurodegenerative disease, they have their own hallmarked proteins which causes the disease. And if we see what happened to these hallmark proteins, so one thing is common in them, that is they all aggregate. Alpha synuclein, it aggregates, it first forms from its native form. When it is in native form, it is important for the system, it is important for neurons, but somehow whatever switch happens, we do not know, but it gets unfolded and then it is misfolded, and because of that change in its folding, it starts aggregating. It aggregates first of all in the form of monomers, then protofibrils, and ultimately it becomes fibrils. Now what are fibrils? Okay, I will talk about it in next slides. So this aggregation is common in all of them. Amyloid beta aggregates in the same form, monomers, protofibrils, and then become fibrils, alpha synuclein, fibrils, everything, Huntington, fibrils. So their aggregation process is same. They all convert their conformation and then become fibrils. Fibrils are the insoluble form of these proteins. Now this could be a target, but there can be other targets also. But let us now, you have already gone through this, the process of Alzheimer's and Parkinson disease. Alan has shown you many different pathways of these diseases. I will just quickly revise it. In Alzheimer's disease, we have APP, amyloid precursor protein, which is a transmembrane protein in the, it is present in the membrane. So this particular protein is in its native form is okay, it does not cause any toxicity, and this protein has some cleavage sites. So if it is cleaved at alpha and gamma site, then whatever product it form is again okay, it does not impact anything, it is non-toxic, and the products are they are soluble and they stay in the cell, they does not cause any harm. But if it is cleaved at alpha, gamma and beta site, so by the enzyme BACE, that enzyme, so then it produces this A beta peptide, a small peptide. Now this small peptide, this has an ability to, this has a property of aggregating. This A beta peptide, it aggregates. It first its monomers from oligomers, then it further form protofibrils, and finally it becomes fibril. Then it is dangerous. So now in Parkinson disease, your alpha synuclein protein, in its physiologically, it is a very normal protein, it is very important protein for the synaptic transmission. It is present in the synaptic vesicles, and when it is present in the synaptic vesicles, its C terminal is slightly exposed outside of the synaptic vesicles. I hope you know what are synaptic vesicles. They carry the neurotransmitters which fuses with the presynaptic membrane and then take the neurotransmitter out from the neurons. So the vesicles have alpha synuclein. Physiologically the alpha synuclein is present something like this, but this alpha synuclein actually help in the accumulation of synaptic vesicles, and it helps in the release of neurotransmitters from this into the synaptic cleft. Alright, Physiologically it is a very important protein, but again somehow something happens, its structure changes. Then what happens when its structure changes? It get partially membrane bound. Its helical portion is now more exposed outside the vesicle, and thus it prevents all the other vesicles to accumulate together. Alright. And then what happens? And then this alpha synuclein protein, its helical structure shifts, converts into beta structure, and then it start forming oligomers, then it start aggregating. It aggregates into, first it aggregates as oligomers, then protofibrils, and ultimately fibril structure, something similar to amyloid fibrils, which is an insoluble protein. And when it is doing all this thing, it thus damages the neurons. So similarly all other diseases in Huntington disease also and in ALS also, superoxide dismutase, this is a enzyme which is practically present in all the cells of the body, but somehow some switch happens and then this protein also changes its conformation and it starts aggregating. So it looks like that aggregation seems to be a initial step, but again Alan posed a question to you, what do you think, this aggregation is the consequence, it is the reason, or it is both? So what answer did you give? Both. So we actually don't know, we don't know. It is a reason, this is a step which seems like it is an initiator, but once it initiates, it promotes further alpha synuclein's aggregation. So alpha synuclein, once it start aggregating, it gives signal to the cell to make more aggregated alpha synuclein. Amyloid beta once form, it start aggregating, it gives signal to the cell to form more aggregated alpha amyloid beta. So it is both, it is a consequence and it is a reason also to promote its aggregation. So what happens, how this aggregation proceeds? Mostly when the protein is in its physiological conformation, it exists in monomer form and it is normal, it does not do any harm, it is actually doing its physiological role, because see, nature what it do is, nature removes anything which is not required by the body. Slowly through evolution, something which is dangerous, which is not required by the body is removed, but we still have amyloid beta in our body. Why is it there? It has a physiological role. Even amyloid beta is also, and in small amount, it is involved in synaptic plasticity, it is involved. So everything has its role, and when they are in their physiological concentration, when they are performing their physiological role, then they exist as monomers. So somehow something happens and these monomers are mostly alpha helical in structure, and then these alpha helical structure, their conformation changes and they actually converts into beta sheets. I hope you understand a protein exists in alpha helical secondary structure and then the beta plated sheet. So some switch happens and the alpha helical structure of the protein, it gets converted into beta plated sheet. Now once the misfolding, we call it misfolding, of the normal physiological protein happened and the protein is now in a beta plated sheet structure, then the aggregation starts. So the first step of aggregation is one monomer will aggregate with another monomer and will make a dimer. Then dimer, another monomer will make an oligomer. So till oligomer stage, it is soluble, the aggregation is soluble. After that, then their structure start making a very you can say you know collagen fibers, collagen fiber ka structure, one collagen then binds to another collagen and then they make such a strong protein fiber which is very strong, and it actually if you see it's you apply force on it, you can't break a collagen fiber. Same way these proteins, when they convert into protofibril structure, they start joining with each other and then they make such a strong fibril structure which is like a very strong fiber that they are no more soluble and then they actually, you know, they cannot stay in the cell, they are thrown out of the cell and they exist in the brain in the extracellular space. So, these fibril structure, if you see here, so fibril structures are what? Beta plated sheets stacked together. They are beta plated sheets stacked together in a very very coordinated and very, you know, nice what do you call that, conformation, and thus becomes insoluble, and they are very stacked together. So, if we see this this particular diagram, so this is actually here we have many different monomers stacked in a fibril form. इसको कैसे देखोगे आप? This is one monomer, this is another, this is another, this is another, and so on. And one monomer is like this. एक monomer ऐसा है, दूसरा monomer उसके नीचे ऐसे stacked है, तीसरा उसके नीचे ऐसे stacked है, and they are stacked like this. All right. So now, if we want to develop a drug which could actually, you know, stop the further elongation of these fibrils, then the drug need to attach somewhere on this monomers. So, most of the drugs, they are, I mean, scientists are working on it, they are developing drug molecules, and if you want to develop a drug molecule which could inhibit fibril formation, then the drugs are made such a way that they bind to the many different monomers stacked together in a fibril conformation. Can you see this? Now, these are different monomers stacked together and the drug binds to them like this. The molecules bind to them like this, and thus it is somehow happening that it is preventing, either it is preventing further fibrillization, or is it is binding to it and then it will take it, it will traffic it to some site where it may be degraded, where it may be processed further, whatever is the role of that drug, it will undergo its fate. So, now again same thing in a brain, healthy brain, monomers exist in alpha helical structure, while they are in oligomer structure and fibril structures are different. Alright. In fibril structure the monomers are stacked on each other, whereas in oligomer structure their binding is different, and so far we are very unlucky, we do not have the X-ray crystallographic pattern or structure available in the PDB of any oligomer structure of these proteins. But it is found recently, I mean yesterday itself Alan showed you about alpha synuclein oligomer which is present in the membrane. That oligomer of alpha synuclein, it can bind, it can come and, you know, get anchored in the membrane, and it act as a pore through which ions can get in and get out, and that pore is switch on and switch off depending on some reason which is unknown. This is a very latest finding, scientists have found it just now. I mean this paper is published very recently in 2005, in which it is found that alpha synuclein oligomer is more toxic than fibril, and these oligomers, they embed in the membrane and they act as some ion channels, and those ion channels open and close on some signal which is yet to be found. Now, the problem is we have X-ray crystallographic data of the fibrils. So far we do not have the structural conformation of oligomers, but it is now found that oligomer stage is much more toxic than the fibril stage. Although fibril stage is also toxic, but the oligomer is much more toxic. So, now as oligomer stage convert into protofibril, now you see the stacking here is different. The oligomer stage structure is different and protofibril structure is different. Now, once it converts into protofibril structure, then it has initiated a, you can there is a initiator. Now, this particular fibril will come start stacking, and then it will lead to formation of fibril. So, this is the first step of fibril formation. So, once a fibril is formed, then this fibril get deposited in the brain and we can easily see the disease in the brain. Alright. It's not moving. Okay. So now as per if we are trying to find a target of this particular drug, particular neurodegenerative disorder, so far studying all these hallmark proteins. So the first thing that comes to anyone's mind is like protein misfolding seems to be the initiator. So protein misfolding occurs and then the whole progress of disease starts. So this protein misfolding is happening because of which reason we do not know, but these could be the reasons. It could be oxidative stress, it could be genetic mutation, then aging. As we age, since alpha synuclein amyloid beta is naturally present in the physiological brain, but these are the neurodegenerative disease are the disease of old people. So somehow aging is also promoting their aggregation. So then we have a system in the cell which maintains the protein conformation. If that system, errata system, is dysfunctional, then we will have more misfolding. If there is high inflammation, neuroinflammation, there is any dysfunction in secretory pathway that Alan told you yesterday, that there is a lymphic system in the brain which is actually continuously removing these amyloid beta if they form aggregates. If alpha synuclein form aggregates, its lymphic system is continuously removing it from the brain. But if there is dysfunction in this particular system, then also it leads to accumulation of protein misfolding, and calcium is also very important. Calcium dysregulation also leads to protein misfolding. Now calcium in the brain neurons, it is very important. It is calcium is present at the presynaptic membrane, and it is a signal which gives information to the synaptic vesicles to release neurotransmitters. So it is important, but its level has to be maintained. If the cytosolic calcium level increases beyond certain level, then it tends to set the neurons into excitotoxic stage, and when a cell is in excitotoxic stage, then the cells start denaturing. Alright, so calcium is also very important. Now if we see calcium and if we see this Erard mechanism, and then the site where proteins are actually folded, so which particular cell organelle come to your mind? Which organelle in a cell maintains calcium in the inside it? Calcium levels? Endoplasmic reticulum. So which organelle is responsible for protein folding? ER. So considering most of the factors associated with ER, so ER seems to be, again, as we are just trying to find a target which could be a good target for designing drug for treatment of neurodegenerative diseases. So ER seems to be a very good target for developing a drug for endoplasmic reticulum degeneration. So it is found that if misfolded proteins or aggregation of protein is loaded in a neuron or in any cell, it impacts its endoplasmic reticulum and puts its endoplasmic reticulum in a stress. So that is known as ER stress. So once endoplasmic reticulum is in stress, then it starts a cascade of events which is called unfolded protein response, and then that unfolded protein response further takes some information to the nucleus, to different sites of the cells, and ultimately if it is continuing in the cell, it leads to neuronal damage, neurodegeneration, because the cell ultimately goes into apoptosis and thus it degenerates. So protein folding, misfolding impacts ER stress. ER stress starts unfolded protein response, and unfolded protein response ultimately lead to neuronal cell death, axonal degeneration, synaptic dysfunction. But when it is beyond one threshold level, actually again any mechanism in the body, any system in the body is actually made for protection. Why do we have unfolded protein response if it leads to neuronal cell death? It is better to remove such a system from the neurons, but actually it is important for the neurons. If there is some minor stress in the neuron, due to some reason, because it is a machinery can go haywire any time. If there are some proteins which could not be properly folded and they are misfolded in the neurons in the endoplasmic reticulum, then unfolded protein response actually tends to correct it. It tends to correct it because what happens, this unfolded protein response gives signal to the neuron to produce more chaperones, to produce something to actually help in proper protein folding. But this till some level. If the stress is so high that it is beyond the threshold, then this unfolded protein response give the signal to the nucleus, now it is beyond our control, just better destroy this cell, and then it takes the cell into apoptosis. So everything is there for a reason, and adaptive unfolded protein response is actually important for neuronal plasticity. I hope you know what is plasticity, but if it is beyond that threshold level, it actually tends to set the neuron towards apoptosis and neuronal damage. Now what is this unfolded protein response? Now when this is a normal endoplasmic reticulum, when it is working properly, the proteins are fine, they are folding normally. So what we have is, we have these transmembrane proteins in the endoplasmic reticulum. This is ATF6, IRE6, PERK. So these are some transmembrane proteins in the endoplasmic reticulum, and they are all bind to, you can see one particular protein on them, which is, here it is mentioned BIP, it is also called GRP78. So this particular protein, it blocks this ATF6, IRE1 and PERK whenever the endoplasmic reticulum is in normal functioning. So the proteins are being formed and they are properly folded, there is no problem in the endoplasmic reticulum. So these particular things are actually blocked by GRP78 or BIP. So as soon as the protein start misfolding, the protein loses its conformation and they got converted into some other structure. So then the endoplasmic reticulum undergoes a stress condition, and in that stress condition, this BIP, it leaves these transmembrane protein and goes and binds to the misfolded proteins. Now actually BIP or GRP78 is actually, it's like a chaperone protein. You know what is a chaperone protein? So chaperone proteins, their main role is to help in proper folding of the proteins. It helps to bring the misfolded, have to correct the misfolded proteins into its actual structure. So GRP78 is one of it's like a chaperone protein, and it binds to the misfolded protein and try to bring them in their proper structure. Either it takes them to their proper chaperone, or if it's beyond control, then it takes it to the Erard mechanism to destroy the misfolded protein. So when the BIP proteins, they leave these transmembrane proteins and goes into the ER lumen, so then what happen, these transmembrane protein, they are no more blocked by GRP78, and now they start their work, they initiate the unfolded protein response. Now what happens in unfolded protein response, all these transmembrane molecule, they further, they go into cytosol, they impact some molecules into cytosol and ultimately cause their impact by influencing the nucleus, and thus whatever was required by the cell that time. If it is an adaptive unfolded protein response, then the cell will start producing more chaperones so that better folding of proteins are made, and if it is beyond control, then it will set the cell into apoptosis. So what happens in unfolded protein response? ATF6, if we look at the ATF6 molecule, so ATF6, when it was blocked with GRP78, it stayed in the transmembrane. As soon as GRP78 left the ATF6 molecule, ATF6 leaves endoplasmic reticulum membrane and it translocates directly into the Golgi. And as soon as it translocates into the Golgi, here the Golgi, it trims it into a active form, and that active form of ATF6 straightway goes into the nucleus, and there it influences the expression of enzymes, expression of mRNA which codes for more chaperones, and those chaperones then go into the endoplasmic reticulum and further help in proper folding of the misfolded proteins. Alright. So, this was all a protective thing. So, PERK on the other hand, when it is bound with GRP78, it stays as a monomer. As soon as GRP78 leaves it, it dimerizes. PERK is actually a kinase protein. Kinase proteins, they phosphorylate. So, this kinase protein, it phosphorylates this EIF2 alpha, this particular molecule, it phosphorylates it, and when it gets phosphorylated, this thing further activates ATF4. Now, this ATF4, it goes into the nucleus, it do many things. It, of course, it helps in increasing the chaperones expression, but it also impacts the apoptosis signaling in the cell. On the other hand, IRE1, this is the most dangerous of all. What it do is, its main role is to cleave RNA. So, first of all, it just cleaves XBP1, it is just an mRNA, and it breaks this mRNA into 2 fractions. So, what its main role is? In adaptive unfolded response, it is just meant to break all the mRNAs, so that it wants to stop more formation of proteins, because now endoplasmic reticulum is in stress, it is not able to properly fold the proteins. So, this IRE1 wants to stop all the mRNA to go into endoplasmic reticulum, so that it wants to stop the protein formation, it wants to stop the translation. Alright. So, it cleaves the mRNA, but sometimes in adaptive unfolded response it is okay, but as it increases, it puts the whole cell, because it is going to cleave mRNA unconditionally, all the mRNAs will be cleaved. So, it impacts the overall health of the cell, and thus this after this it activates, it is not mentioned here, it activates another important signal, that is CHOP, which puts the cell into apoptosis. It initiates the apoptosis in the cell. Alright. So, this is unfolded protein response. Now, so far what you have read is that aggregation is one reason for this disease progress. Now, aggregation of these proteins actually put the endoplasmic reticulum in the cell, or endoplasmic reticulum cell puts these proteins into aggregation mode, and whenever endoplasmic reticulum is in stress, it initiates UPR, and UPR also have this particular mechanism, these many you can see pathways here. So, now just keep them in mind, all the molecules you are going through, they can, and anything can be a good target of a drug, but you have to identify which. So, this is about IRE1, as I told you. Yes, it cleaves XBP1 and thus helps in cell survival, but this has another way of it. When it cleaves ASK1 mRNA, it actually put the cell, it initiates the apoptosis or cell death, it initiates, put the cell into this particular, put it in on this path. So, if we say this, okay. So, again under terminal and unfolded protein response, ultimately all these three, IRE1, PERK2, all of them finally leads the formation of small vesicles, which ultimately become the autophagosomes. So, as Ellen is telling you about that, under these neurodegenerative disorders, mitophagy happen, phagy happens, and ultimately the cells undergo autophagy. So, if we see the mitophagy, phagy, autophagy, if we go upstream of it, then it seems like endoplasmic reticulum and unfolded protein response seems to be an initiator of these mitophagies and phagy. But again it's hanky panky. It's always like sometimes the Answer is it's egg or the hen first, because sometimes it looks like the ER-phagy is initiating autophagy, but sometimes it's like mitophagy initiates or it gives signal to the endoplasmic reticulum to make more such vesicles and bring more molecules into mitophagy and ER-phagy. Now if you see, like the thing that I was telling you, now if you see all the diseases, different neurodegenerative diseases, and if we see the hallmarks of those diseases and their impact on endoplasmic reticulum, so there are many studies, and if we try to sum up the studies in one such particular diagram, so we can see that if you see here, it is found that if there is alpha synuclein load in the neurons, or there is Huntington load in the neurons, or VAP-B load in the neurons, it influences endoplasmic reticulum signaling, and it influences unfolded protein response. It actually enhances it, and thus give the signal to the nucleus to start the unfolded protein response expression of mRNA. Alpha synuclein Huntington also influence the export of proteins from vesicle into the Golgi. It is found that amyloid beta, prion and Huntington, it impacts the calcium hemostasis. So far, role of alpha synuclein in calcium hemostasis is not found, but amyloid beta can impact the calcium hemostasis, and majorly it increases the calcium load in the neurons. Same way it is found that Huntington and tau protein, not amyloid beta, tau and SOD1, it influences the Erard mechanism. Erard mechanism, now what is this? This is again a quality control check of endoplasmic reticulum. If there is a load of many misfolded proteins in the endoplasmic reticulum, and the chaperones are not good enough to bring them back to their actual structure, so those misfolded proteins, they are sent to the Erard mechanism, where those misfolded proteins are destroyed and removed from the cell. So this is a quality control check of endoplasmic reticulum. So these proteins, Huntington, tau and SOD, it impacts, it slows, it reduces the activity of Erard mechanism in endoplasmic reticulum. And it is also found like alpha synuclein aggregation, when it starts, SOD1, prion, and this aggregation once it starts, it impacts the folding of the proteins in the endoplasmic reticulum. So again the same thing, whether it is the folding, misfolding that initiated alpha synuclein aggregation, or alpha synuclein aggregation which initiated the folding, they both are interconnected. Alright. So it is found that once we add aggregated alpha synuclein to the neurons, it turns the switch on in the endoplasmic reticulum to make more unfolded or misfolded proteins in the endoplasmic reticulum. Alright. So these are the things that happens in the endoplasmic reticulum under different disease conditions when their hallmark proteins are exposed to them. Now till now, what do you think could be the best target for therapeutics of neurodegeneration disorders in ER? Now just it's some point for you to ponder. What do you think it could be? If we somehow manage the Erard mechanism, can it be a good drug target for this particular disease? Yes. Which is the best? Splicing. Okay. Splicing, for splicing what we have to do? It is the IRE1. So what we have to do is we have to inhibit IRE1. I mean, if we want to make IRE1 as a drug target, so what we are going to do is we are going to block it so that it should not be able to splice any mRNA. So will that be really good target for this for this disease management? What do you think? What others think? Just ponder what you have. I have told you some mechanisms in it. So now what could be the best way to treat it? Okay, what if we block it? Absolutely. Very good. Hmm. Hmm. So you are saying that we should not put more effort on Erard mechanism because we should, this is something which is coming after the protein is misfolding. So we should look something here, that something which should stop protein misfolding. Okay. So what do you think should be the target? Think. There is no wrong answer. There is no wrong answer. Anything can be a protein, I mean drug target. Chaperones, okay. So these chaperones, okay. If we can alter, what do you want? We should increase their activity? Okay. If we increase the amount of GRP78 and then it will block the unfolded protein response. Okay. So now what will happen? You have misfolded proteins, alright. You have some misfolded proteins, not much, and you have increased GRP78 which will block ATF4, PERG and IRE1. Now what will happen? The misfolded proteins are like that only, they will stay in the endoplasmic as it is, and you have stopped the adaptive UPR. Adaptive UPR was actually good. It would have actually worked by enhancing Erard mechanism, by enhancing other chaperones and by enhancing other mechanism to maintain protein folding structure. So what we did is we just stopped one step. There is no wrong answer, but I will tell you what, everything has some drawback. The point is this, every strategy has a drawback. That is why we are not able to find a drug so far. So this way, but the misfolded protein load is still there in the endoplasmic reticulum. We just stopped UPR, but some UPR was also required.
Ma'am, one of the safe approach to it will be managing the calcium homeostasis, because all other mechanisms have some intrinsic role, they have physiological role. And if we are going to turn off the switch permanently, then in case we actually need some unfolded protein response for some other toxic proteins, etc., then it will be harmful in that way. Also, now the relationship is pretty bidirectional. We actually don't know seeding is leading to this or either this is leading to seeding.
Yes.
So,
Okay, yes. Okay, so you are saying, you said many things, but I'll just pick one thing. You are saying if we could stop the calcium influx in the cell. So now what about what is the role of calcium influx? Why we need calcium in a neuron? As I said, it is important. Yes.
targeting the homeostasis, not like.
Okay, how could we maintain hemostasis? Very good. How could we actually maintain hemostasis? We can't block the channels because channels need to work. We need appropriate amount of calcium in the neuron. We just can't stop it, but we need to maintain its hemostasis. Its level must be maintained. How could it be maintained?
There are some indirect mechanisms, for example, inhibiting the TRPs channel inside the cell. Or actually, it's quite interesting because if we inhibit amyloid beta or tau or whatever protein, it also indirectly inhibits the calcium because there are multiple steps.
So yes, So now you yourself gave an answer. We cannot just inhibit the channels because they are important. They are actually maintaining the calcium hemostasis. We are, this hemostasis is actually lost because of the protein misfolding. So now you gave a target, that is amyloid beta aggregation and or alpha synuclein aggregation. So can we find anything here as a good target? This is the mechanism. This is the mechanism, and everything is actually important. We can't stop one thing for the other and other. But yes, although I may be wrong, but so far, Hanji.
Can we increase the receptor, like sensors like IRE, like instead of lesser of IRE1, if you increase the receptor, IRE1 protein. So in this one, like you're not even stopping anything, but you're increasing this mechanism. Like I'm thinking in case of more transcription factors and chaperones will be useful.
So you are saying increase the expression of IRE1 only or chaperones also?
Only for IRE1.
Only for IRE1.
You are saying UPR is not able to press, like it has a threshold. So I'm saying increase the IRE1, so the threshold might increase. Like you increase this threshold point. Let's say
No, if we increase IRE1, what will happen? We will destroy more mRNAs, and but destroying mRNAs which are required for vital functioning of the cell is also not good, isn't it? So we have to actually limit it. We don't want to destroy all the mRNAs in the cell because there are many other vital functions like glycolysis, Krebs cycle. These things must be, these things must continue. Their enzymes must be continuously synthesized. So we should have some, translation also occurring in the cell, even though the cell is in stress, I think. It's good. It's good. Just, we should think. It's very good. I'm very happy that you are thinking on some terms. Give a hypothesis and then try to find its limitation yourself. Okay, if we'll do this, then what could happen? Oh, these things can happen. If we'll do this, then what could happen? These things can happen. Give your hypothesis and find its limitations. This is how your brain should work. When you are studying any disease, when you are studying its mechanism, you should see every step, every pathway's metabolite as a drug target, and then see that if I target it, what benefit it could give, what drawbacks it could bring with it. This way we can actually narrow down to what is best for the treatment of the disease. Alright. So far everything is so complicated, and anything can be a target, but anything can, they have their own limitations.
We can also use microRNAs here, which can suppress these proteins.
MicroRNAs to suppress protein aggregation.
So that
Why you want such an expensive thing? Okay. Okay. I thought you were going to say microRNAs to suppress gene expressions. There are treatments, there are studies going on in which microRNAs are being explored to manipulate gene expressions. So again, that becomes a genetic therapy. Translating it is a challenge, but yes, there are many papers you will find in which microRNAs are also being explored for the treatment of neurodegenerative disorders. So it's okay, I think you have thought, I have given enough food for your brain. We can move forward. Now let's talk about the chaperones in the endoplasmic reticulum. So now there are many resident chaperones in the endoplasmic reticulum, and their role is to give proper folding to the protein. One of them comes under a class of heat shock proteins. So the heat shock proteins like heat shock protein 70, it helps in folding of the protein, and it is dependent on ATP molecule. When it is bound to an ATP molecule, it is in an open form, and then the protein comes and get attached to it, and then ATP is removed, then it tightly binds the protein, whatever changes in the protein structure happens there, and then ATP is again regenerated, and then the folded protein comes out of the HP70 chaperone. So sometimes HP70 does not, cannot fully give structure to all the proteins. So partially folded protein then goes into HSP90, and HSP90 is very important. It gives, mostly it ends up all the protein in their proper folding structure. HSP90 is actually a dimer, and the protein gets attached in the cleft between these two dimers. ATP molecule is consumed to give the protein its proper structure, and finally a proper folding of protein is achieved. Alright. So there are other chaperones like calnexin and calreticulin. So these chaperones are basically involved in glycoprotein structure. So they are mainly involved in adding the carbohydrate group on the protein structure. So if you see most of the these transmembrane proteins, they all have glycoprotein, they all have a carbohydrate group attached to them. So calnexin and calreticulin also have an important function. So this is just a basic idea about the chaperones, and these chaperones are natively present in an endoplasmic reticulum, and they are doing their work. Whenever the misfolded protein burden increases, their expression increases to maintain the misfolded protein burden. Okay. So there are the molecular chaperones are the chaperones which are natively present inside the endoplasmic reticulum, and how they work is, due to any stress condition when the proteins are unfolded or misfolded, these molecular chaperones, they come and bind to it, and then they form a protein complex with it, and then they turn them into their functional state. If these chaperones are less in number, or unfolded proteins are more and chaperones are less, then these proteins, these unfolded proteins, they start aggregating, and thus they lead to this particular disease condition. Now apart from molecular chaperone, there are other class of chaperones, that is chemical chaperone and pharmacological chaperones. They are not normal resident chaperones in the cell, they are drug molecules, chemical molecules, which have ability to do the same thing as the molecular chaperones do, and they thus become a very good candidate for the treatment of this disease, to actually block the aggregation of these molecules. So if I see all the mechanism, not I, many other scientists, if they see this mechanism, so they found that if we could somehow reduce the aggregation of these proteins, then we can treat this particular disease, and chaperones become a very good method of treating these aggregations. Now we cannot make the molecular chaperone as targets, because molecular chaperones are again proteins and very big proteins. We can't use protein as a drug. We need its alternative, and we have many alternatives so far. Since last many decades, scientists are working on it. So we have chemical chaperones, we have pharmacological chaperones, and these molecules, they actually mimic, they work in the same way as the molecular chaperones. So now molecular chaperone is clear to you. What are chemical chaperones? They are some chemical moieties. They are not specific, they just don't bind one particular protein and give it its structure. They act same as the molecular chaperone. They help in giving the structure, protein folding structure, and help in its proper folding non-specifically. Then we have pharmacological chaperones. So these chaperones are some molecules which are specifically designed to fold specific proteins, and there are many reports in the literature where natural compounds from like ginkgo biloba, curcumin, so these compounds are also found to have ability for enhancing protein folding. and they seems to be quite specific, as these extract like TCM, it binds specific to some particular proteins. So, if you see the mechanism of action of these chaperones, it is clear. The chaperones, what they do, they directly bind to the misfolded proteins, and they also reduces the endoplasmic reticulum stress, and thus impact the UPR and reduce the burden of aggregated proteins, and thus treat the disease. So, once endoplasmic reticulum is maintained, it also will reduce the burden of, it maintains the calcium hemostasis. So, these are certain, there are many chemical chaperones known so far. These are the most popular one: TMAO, trehalose, TUDCA and PBA. So, these are some molecules. Now, you see their structures, they are not so complex. So, these molecules, they actually act as chaperones, and they are very helpful in maintaining proper protein folding. So, if we see their mechanism of action, so I will skip this part. So, they are, I told you, there are some of them are specific, some of them are non-specific. I will straightway come here. Now, trehalose and glycine. So, these they are osmolytes. Osmolytes directly attach with the misfolded protein and help in its proper formation. Alright. Osmolytes has just one role, has just one mechanism. This is mentioned here only. So, I will just show you in this slide. So, they have just one mechanism, that is giving the protein its proper structure. So, they do not have any other role. So, osmolytes are very good in giving protein structure when we use them in vitro, but as soon as we use osmolytes as a drug in the cells or in the animals, it is very difficult to translate them, because they do not have, they are not specific, they do not reach to the target site, they do not have any other mechanism by which they can help in the treatment, and an osmolyte, one osmolyte molecule should actually reach the misfolded protein to cause its action. So, it is very difficult to translate it, but osmolytes, when you put it with an unfolded protein, it helps in its proper folding and it prevents its aggregation. So, apart from that, these two molecules, PBA and TUDCA, they are found to be very good chaperone molecules. So, TUDCA and PBA, there are certain reports which says that they directly bind to the unfolded protein also and prevents its aggregation, but they have multiple roles. They are also involved in reducing the inflammation. They are also involved in, they goes into the nucleus, like TUDCA, it binds to this ferronoid X and does impact the expression of certain genes in the nucleus, and your PBA, it binds to HDAC, histone deacetylase, and it impacts the expression of some genes in the nucleus. So, they have multiple roles. So, PBA, not only that, PBA have another role. What PBA do, if we have unfolded proteins in the endoplasmic reticulum, they are not encapsulated in the vesicles, only the properly folded protein molecules get encapsulated in the vesicles and are removed from the cell. But when PBA is present, it encapsulates the unfolded proteins also and removes it from the endoplasmic reticulum, thus reduce the load of misfolded protein and endoplasmic reticulum. So, PBA has multiple roles. So, considering all these properties, now if we see the molecular structure of TUDCA, it is very complex, and this seems to be simple and it has many other properties. So, we can zero down on the molecules, So, in our lab we zero down on 4-PBA. Now, 4-PBA is a molecule which is a very small molecule, it is a short chain unsaturated fatty acid. This is actually an FDA approved drug. It is used for uric acid disorders, not for neurodegenerative disorders, but for uric acid disorders. It has a very high LD50, so it is not toxic at all, and there are many studies which says that it has good chaperone activity. So, we studied its activity in in our lab also. This is my PhD student who studied the role of PBA in excitotoxicity. We induce excitotoxicity by giving them AMPA, and what we found was that PBA actually it reduces endoplasmic reticulum stress in excitotoxic animal models. It maintained calcium influx of endoplasmic reticulum stress, and it also improved the cognitive abilities of the animals in excitotoxicity. So, it seems to be a very good lead target. Now, if we consider aggregation as a target and we want to prevent aggregation of molecules, and then PBA becomes a very good lead molecule. So, if it has such a good property, why it is not yet translated as a drug? So, this is the outcome of that study. I will just skip it because we are short of time. So, now why we could not translate it? So, the reason is, although PBA has a very good activity, it is very effective, but the dose which caused this efficacy was around 200 mg per kg weight in animals, and if we translate this drug to humans, then it will come out to be around 3 grams per day. So, a drug at this high dosage, it is not feasible to be ingested by a patient and to be used by a patient. So, its dosage is high. So, the first and foremost step which we need to do is, we need to develop a molecule. Now, if you see my first slide, we have selected a target. The target is aggregation. Now, we found a lead molecule which actually stops an aggregation, that is PBA. Now, we will improve it. Now, PBA needs improvement because its dosage is very high. We need to make it in such a way that it shows the same efficacy with less dosage. So, how to do that? Yeah, we have to make some new molecules, we have to make new derivatives of PBA and then test them. So, how would you do it? You will start randomly making derivatives? No, you should have some basis. Yes?
Take some molecules that do not produce any other side effects, like bioequivalent. So we should modify the molecule as such that it has no side effects.
Okay, how? This is your PBA, how will you modify it? This.
So ma'am, by analyzing the structure activity relationship, then keeping the groups same which are important for their management, and
Yes.
deriving a small molecule.
Yes, we need to have a base, we need to have some information that we can't, if we randomly change it, yes we can, when we do not have any information, we have to randomly change this molecule, we have to randomly add many different groups, but if we have some idea, then it helps your study. So, there is a study by Memory in 2012. So, he gave a very good, good outcome of his studies. He said that we can enhance the activity, anti-aggregation activity of PBA if we increase the aliphatic chain in PBA, if we add a methoxy group at the para position in the PBA, it enhances its anti-aggregation activity, and if we add more electron clouds, benzene rings to this molecule, it enhances its anti-aggregation activity. So, these findings were given by Memory, and on the basis of these findings, now we can modify this this molecule, and we can add chains and keep on increasing. We can add benzene group, we can add methoxy group, and so far we can change. Now, we know what changes we need to bring in this molecule to enhance its activity. So, what we did, some of our students sitting here in my laboratory, they made a huge library of this modifications. They made a library of around 30-40,000 molecules, and then they tested its ability first of all in silico. So, how this is done? So, now we can't do these changes in each molecule and then test it directly on the animals or on the proteins. It will be very time consuming and it will be very uneconomical. So, to bring it to make it more economical, we use these in silico tools. So, what these in silico tools are, we design a huge library in silico. So, after lunch we will have a session of this in silico drug designing. How many of you have experience of in silico drug designing? No one. So, this next session will be really helpful to you. So, what we do is we make a huge library of molecules. We should know what changes we can actually do it. If we do not know, then also it is okay, we randomly change the molecule. So, after that we test the binding efficacy of those molecules with the target protein, how strongly that molecule binds with the target protein, and then we then those molecules which showed very good binding with the target protein, they are tested for their ability to become a drug. Like sometimes, like if you see this TUDCA molecule, this is a very good molecule, but it is very complex. It cannot actually be translated as a drug. Why? Because it will have low, it has low blood brain barrier, it cannot cross the blood brain barrier. So, if our drug cannot go into the brain, then it is of no use. So, we need to make such molecules which has good absorption. They should be easily absorbed in the intestine, they should have low toxicity, they should be able to cross the blood brain barrier, and they should have other best properties to actually become a drug, and that also is studied in silico using ADME analysis. So, you will study that today after lunch, we will have a session on that also. So, once we screen all the library, we see which molecules best bind with your structure, then we see which molecules has best property to become a drug. They can cross the blood brain barrier, they have less toxicity, they are less absorption, they have more absorption. So, those molecules are then selected and synthesized in the lab, alright. So, when we synthesize these molecules in the lab, normal chemistry reactions are done to synthesize them chemically in the lab, then their structure is validated by spectroscopic methods. Here I am just showing FTIR, but we do NMR, MS, other methods also. So, the structure is validated and then they are tested in vitro and in vivo. As I have shown you in the first slide, we select the target first, then we select the lead molecule. Lead molecule is PBA, it has some problems, we need to improve it. We improved it, we are now improving it. We improved it through in silico studies. We synthesize these molecules now, and now we will test them on in vitro platform. In vitro platform in our case will be what? We are testing them on, we are testing their ability to impact aggregation. So, our in vitro platform is anti-aggregation ability. So, now what we will see is how well the molecules are binding with your target protein in vitro directly, and are they actually preventing aggregation. So, for that we will straight away take either amyloid beta molecule protein, peptide, or alpha synuclein proteins and expose them with your drug molecule, induce aggregation in them and see whether your molecule is actually inhibiting the aggregation or not in vitro. And there are other methods also, instead, like we are making a non-specific chaperone molecule. So, it is not only impacting alpha synuclein or amyloid beta. So, we can also study them on any other protein. We can take BSA, induce aggregation in them and test our molecule whether it is inhibiting the aggregation in them or not. So, this is in vitro studies, and after that we take it to the animal. In animals we induce the disease. We can either chemically induce the disease. I will tell you about this animal models and animal studies in my next talk to you on, which is on Friday. So, I will just give you a brief idea. We induce the studies either chemically, like for Parkinson disease we give rotenone to the animals, this induces Parkinson disease in them, or we can take transgenic animals. Although now transgenic animals are considered better model for neurodegenerative diseases. So then we check our drug molecules whether they are actually helping in reducing the this disease load in them by various parameters. And after we complete all these steps, so we then only then can narrow down on a molecule which has crossed the preclinical 5 steps. We have a molecule which could reduce this disease burden in animals. That molecule can go further in the clinical trials. All right. So that's it. Thank you. Any questions you have? Yeah.
As you said, that you are checking the binding of the molecule to various peptides. So, for example, amyloid beta has several forms. For example, amyloid beta 25 to 35 peptide, 40 peptide, 42 peptide. So, we have to test individually in every peptide that which one has better binding.
Why should we use, I mean not all peptides are having, you know, aggregation potential. It's 142 is the most prone to aggregation. So we should make a drug which could actually bind to the most aggregating peptide.
But in the animal forms, we don't use the 40, we use other forms like 25 to 35.
No, we can use 142 also. We stereotactically can inject this 142. We have that model only, and that initiate the aggregation in the animals. In animals, we use 142 also. Yeah.
You did the study after your in silico screening. How many molecules did you arrive at?
Okay. Actually, after in silico studies, we get, I mean, we just put them in order of their best binding ability, and then we check their ADME scores, their ADME properties, and we have to see the feasibility of their chemical synthesis also. So it all depends, the feasibility and all. Sometimes you can synthesize 15 molecules, you can synthesize 25 molecules, you can synthesize 4 or 5, it varies from the structure to structure. So in our study, I think we have zeroed down to 4 molecules. 4 molecules.
And the in vitro was done with all the 4 molecules.
All the 4 molecules, yes. In vitro study was done with all the 4 molecules. Any other question?
So, yes, definitely one way is to have all the modifications done in the molecules or find molecules. Or else, one way to do it also be having combinatorial therapy where 2 molecules or 2 compounds essentially enhance each other's effects, or not each other, the other one's effects that we require. So,
Which is, you are asking if we give a combination therapy, is that better, or we should modify the drug molecule? Okay. For combination therapies, this is my opinion, like they should have different targets. Like both the drugs should not be acting on the same target. If they act on the same target, then they are competing with each other, isn't it? So now our target is, when our target is 1, that is aggregation. So here we, I mean, we cannot have a combination therapy. But like if we want to target, let us say hypothetically, we want to target aggregation and we want to target, let us say, calcium hemostasis, then we can use a combination therapy. But again, you know, for combination therapies, it gives good results. I mean, you can find results sometimes, this is happening, this is actually it's giving a synergistic outcome. But if we want to see the mechanism, sometimes some mechanisms are interlinked, sometimes some mechanisms are not interlinked. So it's sometimes very difficult to pinpoint that what could actually have happened. But there is, I mean, research are going on, people are working on it. But when you are trying to find a new molecule, when you are trying to find a new drug, I think at that time you should focus on one target. So once you are confirmed that your one target drug is best, then you should try different combinations with it. And when you are doing combination therapy, then your target should be separate. They should always be different. Never use one target as, I mean, never use two drugs acting on the same target.
How about if for this example, we are targeting aggregations, so aggregates are going to be broken down. Is that what we are looking at, or are we looking at stopping the formation of aggregates?
Very good. So this is very important. So Alan has shown you some drugs, monoclonal and antibody based drugs like Wemby. So their mode of action is same. They are going to stop the aggregation of these proteins. Like Wemby is going to stop aggregation of amyloid beta, and so far we don't have any drug for alpha synuclein. Yeah. So those drugs are actually effective when they are given at the early stage of the disease, because these molecules actually they prevent the aggregation. But once the protein or peptides they have actually come in the fibril form, it's very difficult to break them. It's very difficult to break them. So this therapy can work if we start giving them at an early stage when these molecules have just started aggregating. Prior to, once a fibril is formed, yeah, once a fibril is formed, it's very difficult to break a fibril. Fibrils are very stable structure. We can actually prevent its further growth, and we can stop the aggregation at the initial step.
And would it be helpful to say further that when we are stopping aggregation at the error stage, we know early stage would be something has gone wrong and aggregation has started, which we are trying to stop. So when aggregation seems stopped, along with it we also need mechanisms that clear out the area to work as best as they can, so that those broken down aggregates which are somewhat could be harmful.
They are not harmful. That's what limbic system of the brain do. It's cleaning the brain, continuously cleaning the brain.
Now, many times both of the systems go wrong at a similar time frame. So
With the age, this limbic system also gets low. But once your once you have given a drug which binds to your monomer and is preventing its aggregation, so now that that monomer will not whatever harm the cell, it won't be able to do that. So what will be its fate then? It will recognize it from foreign and system in the body to remove the debris. I think anything else? Any further query? No. So I really stops in the afternoon, but problem can drop or not solved. So don't leave just now. We'll tell you something about in software in your, you might have brought your laptop. So after lunch we'll have on drug designing, where we'll just brief idea how could we design and how could we work on it. So just Shiva just इनको बता देना, ठीक है. So now we can meet 2:30 sharp. Please be 2:20. Start the session at 2:30.